So first, I would like to thank organizer to give me such a good opportunity in front of big blackboard to share my recent result. But I don't use a blackboard. Today I'm going to talk about the cell alignment at the leading edge of the bacterial swarm. The experiment is mostly done by this my master student, Kakeru. Yes. Now it starts the movie. This is a bacterial swarm. Okay. So, yes. So first, I will introduce you a little bit about the bacterial swarm. So when I spot a drop of bacterial culture onto the agar plate and wait for maybe 16 hours, and bacteria grows and spread over the agar surface. And this is the experimental result with the wild type. This is very distinct from the usual colony observed during cloning or something. This is an experimental result from a swarm deficient cell. So these colonies are thick and dense, but this one is thin and big. So during swarming, cells are motile and elongated. They are lined. So the bacterial swarm could be a model system to study the spatial organization and active matter. So our model organism, it's Vibrio algonalyticus. Usually it is in the seawater, but it comes to the surface of the solid, like a seaweed or plankton. So it has a morphological change. Cell is highly elongated, and it generates large amount of lateral flagella. This is a driving force on the solid surface. Here, this is a movie of the single cell movement in the swarm. These cells express GFP in the cytoplasm, but the rest are non-fluorescent. So you can see the single cell movement in the swarm. Yes. So you notice that these cells are very, very flexible. It's not like a usual bacterial swarm, E. coli or Bacillus, and it's rather similar to the active filament, like actin or microtubules. And this movement is regulated by a cytoplasmic regulator KY, and the KY, okay, KY, yes. KY is phosphorylated by the kinase located in the cell pole, and then it diffuses into the cytoplasm, and it is bound to the flagella motor. It results in the slowdown of the flagella rotation. And thus, the KY deficient cell, delta KY, we call it, moves very fast on the agar surface, but it failed to swarm like this. This is the picture I showed you previously, swarm deficient cell. Even though the KY phosphorylation is regulated by chemotaxis pathway, but the previous study using E. coli demonstrated that the chemotaxis itself is not required for swarming. KY is only required. Okay. The central question in my presentation is how the cells align the orientation at the leading edge of swarm to swarming. This is actual swarming movie. Okay. So first, we characterize the cell orientation at the leading edge. Okay, this is wild type's picture. So we set the coordinate system like this, and the y-axis is parallel to the direction of spread. And in the right, we obtained orientation field as a function of the y. And in dark blue one, this is exactly at the leading edge. You can see the sharp localization at the 0 and the pi radian that correspond to the x or minus x direction. So this means leading cells are oriented perpendicular to the direction of the spread. This is a bit counterintuitive because as I showed you, the cells move along its long axis, but they are oriented perpendicular to the direction of spread. I'll come back to this point. And then this sharp localization relaxes as the Y decrease within the 5 to 10 micron, within the size of a single cell. Okay, then we characterize also the delta key Y. Maybe as you can see here, the cells are oriented toward the direction of the spread, like this. And actually, there's no sharp localization at the 0 and the pi radian. And also, we note that these leading cells are stuck. You can see these leading cells are stuck, but the trailing cells are moving like crazy. So these leading cells act as a late to prevent trailer cells move upward. Okay, this kind of behavior is also observed in the wild-type cell. So sometimes wild-type cells come to the leading edge, but they are misoriented. I will start the movie. You will see 3 leading cells come to the center. No, this one. This one? Yes. And stuck. I will continue the movie, and the 2 of them show the reversal and go back to the swarm. And the last one is swallowed by the swarming wave. Yes. Go back. And the last one is swallowed and helped. Okay, so we suspect that this kind of behavior is due to the hydrophobic interaction between the surface and the cell. So thus we applied the surfactant to reduce the surface tension between the cell and surface to see the change of swarming in delta QS cells. And we measured the ring diameter of wild-type delta QI in the absence of surfactant. It showed a small ring. But once we add the surfactant in the agar plate, it spread more than wild-type. So the swarming is recovered. So from this, we conclude that misoriented leading cells are stuck due to the hydrophobic interaction between the cell and the surface. So the proper orientation at the leading edge is required for successful swarming. Okay, so first question, the answer for the first question is leading cells are oriented perpendicular to the direction of spread. And then next question is how? Oh, sorry, how? Okay, to this end, we tested the hypothesis. We have 2 hypotheses. So one is a compression-induced alignment. This is previously proposed theoretically and was demonstrated as valid in the case of actin filament during cytokinesis. And trailer cells push the leading cells, and the leading cell reorient. Another hypothesis is a dynamic turnover at the leading edge. So trailer cells move toward the leading region and return to the trailing region. So we sought to test these hypotheses in experiment. So we decided to do fluorescence recovery after photobridging. So we image the GFP expressed cells. And then we applied the strong light in the certain region of the cells and to breach the GFP. If the compression dominates, the breached region moves along the leading edge. But the turnover dominates, the bacterial breached region becomes mixed by the non-fluorescent and the fluorescent cells. Okay, so this is the experimental result. Okay, white rectangle shows the breached region. I will start the movie. Breached and comes from the trailing region and also from the lateral region. So this suggests that the dynamic turnover of the leader cell is happening during swarming. And we measured the fluorescence recovery time constant. It was 40 seconds. And also, we measured the dwell time at the leading edge. And this is also 40 seconds. So the dynamic turnover of the leader cell is around 40 seconds. It's quite fast. Okay, then we studied the cell trajectory in order to understand the dynamic turnover. I will start the movie of the wild type and delta KY at the same time. No, this. Yes. Okay. As you notice, the delta KY has higher motility. It moves faster, and also its movement is persistent compared to the wild type. The wild type is very flexible and show the frequent turning. So to characterize these dynamics, we computed the curvature of the trajectory like this. But now we don't need to take care so much of the sign of the curvature. The curvature could be positive or negative. It depends on the left-handed or right-handed. So we don't need to take care. So we took the absolute value like this. And then we took the get ensemble average in wild type and delta KY. Then we found a 5 times higher value of the trajectory curvature in the wild type compared to the delta KY. So this means a low persistence movement of the wild type cells. Okay. This result and the orientation at the leading edge suggests that the low persistent movement promotes cell alignment at the leading edge. This is just idea. Okay. So trailing cell come here. In the case of wild type, it shows the frequent turning like this and go back. But this is not the case of the high persistence mutant, like delta KY. It comes here, but it doesn't rotate, turning. So it's stuck. So this is just idea, but this is supported by the surfactant experiment. Yeah, as I told you, the addition of surfactant result in the spread of swarming, but the trajectory curvature doesn't change. So the cell movement is all persistent, even if we add a surfactant. It's just a problem at the leading edge. So this is the orientation distribution in the delta KY in the presence of surfactant. So it's roughly localized to the 0 and the pi radian that correspond to the perpendicular, the angle perpendicular to the direction spread. Okay, so we conclude that the leading cell undergo turnover, moving from and returning to the trailing region with low persistence movement. Okay, so next we asked how the KY contribute to the turning behavior. First, we sought to moderate the intracellular concentration of the KY. To this end, we induce KY into the delta KY cell. By changing the inducer concentration, we can change the KY concentration. We check this the presence. We measured trajectory curvature here, and the trajectory curvature doesn't show the monotonic increase as a KY concentration, but shows a peak here, 0.3 millimolar. And interestingly, at this condition, we saw the spread of the swarm. So there is a strong correlation between the ring diameter and the trajectory curvature. And so this supports the idea that low persistence motion enables reorientation that is essential for the swarming, previously I showed. Okay, then we studied the relationship between the turning and the cell bending. So again, we computed projective curvature, but as a function of end-to-end distance of the cell, as a proxy of the cell bending. Okay, cell bend as end-to-end distance is like this. Okay, in the left region, the end-to-end distance is small. This means the cell is largely bent. In this case, the trajectory curvature is higher. This means the cell tends to turn. But the end-to-end distance is larger, the cell rather moves straight because the lower value of the trajectory curvature. So from this way, okay, cell bending promotes the turning behavior. But we notice that this is only the case of Y type, not for the delta KY. Okay. KY phosphorylation is localized cell pole. So this is a fluorescence photograph of the KY GFP. So you can see sharp localization at the cell poles. This is because the highness of KY is localized at the cell pole. And so the phosphorylation of the KY is localized at the cell pole, and once it is phosphorylated, it's diffused into the cytoplasm and to bind to the to be bound to the flagella motor to make the being rate slow down. And in E. coli previous studies, regulation of the flagella rotation by KY proceeds sequentially from the motors near the cell pole. So if the motor is very close to the cell pole, it's change the spinning rate earlier than compared to the other one. Okay, based on this experimental result and this experimental fact, we hypothesize the compression-induced buckling. So as I told you, the KY phosphorylation is localized at cell poles. So we could expect the inhomogeneous distribution of the KY in the cytoplasm. Maybe in the cell pole, the phosphorylated KY might be a lot compared to the Y type. This results in the velocity gradient along the long axis of the cell. Because there are many KY here, and it makes the spinning rate slow down, but it's not the case of the here. And this velocity gradient induces buckling and turning. Okay. Here it's slower, it's faster. The tail head push the head region and backwards. Okay. So this idea is kind of supported by the analysis of the cell bending dynamics. So we computed the curvature of the cell shape. Now I didn't take the absolute value. We took the kymograph of the cell curvature. Okay, the left-hand side, it's head, right, it's tail, and the y-axis time. Okay, so now the cells moves forward, and the curved region, curved region is red or blue, propagate from the head to the tail. The bending region propagates, so like here to here. But interestingly here, the cell exhibits reversal. Now tail is head front and head is rear. Then the propagation is also flipped. The propagation starts from the tail to the head. Okay, so the bending propagates from the front to the rear of the cell. So, but we sought to test this hypothesis in experiment. So to this end, we use a constitutively active KY. So this is an active form mutant, This means we don't need, it doesn't require the phosphorylation. It always be able to be bound to the fragile motor. And also we checked distribution of this KY is homogeneous in the cytoplasm. So there is no inhomogeneity of the distribution. Okay, and then we, okay, this is a movie. Okay, okay. Okay, so this is a movie of the constituent CAQY in the delta QY cell. So okay, it's kind of similar to the movement of the delta QY. And actually, we measured the trajectory curvature, and this is almost same as the delta QY. And then, okay, we check the relationship between the trajectory curvature and end-to-end distance. And we found that the turning behavior promoted by cell bending is not fully supported by the CAQY, as shown in brown. And CAQY failed to promote the alignment at the leading edge. This is the orientation distribution at the leading edge, and also the fail to swarm. So we concluded that the localized activation of the KY is required for the low persistence movement that is essential for swarming. Okay, to sum up, the leading cells are oriented perpendicular to the direction of the spread, which is essential for swarming. And the leading cell undergo turnover moving from and returning to the trailing region with low persistence movement. This movement is promoted by cell bending. The bending is driven by the localized KY phosphorylation. Thank you.
Thank you very much. I'm curious, since your constitutively active KY is very similar in behavior to your delta KY, and you mentioned that you need KY, but you don't require chemotaxis or swarming. Then what about KY is doing something different than binding to the flagellar motor that's driving this behavior?
You mean the...
In other species, apart from E. coli, that have multiple chemotaxis systems, there's evidence that KY has other influences besides simply altering direction of rotation of the flagellar motor. So I'm just wondering, throw out your old ideas of interacting with the flagellar motor and think about KY in a different context.
Okay. At least with...
We can talk about it over coffee.
Well, wait.
Okay.
So maybe I'm a bit confused. You mean the KY? KY is required in...
What? It's a longer discussion. Yeah, let's have this over coffee. But I just am curious that there's a role for KY here that doesn't involve its interaction with the flagellar motor.
Interaction with the flagellar motor. The interaction with flagellar motor is important, also in the E. coli swarming. But okay, I'll explain the previous study imprecisely. Okay, what they found is, okay, in absence of KY, swarm doesn't spread. But in E. coli, it's easy to remove all the receptors. But still, even if the cell has KY, it succeeds to spread. So the chemotaxis itself is not required. And even if the KA is not present, not localized, KY is autophosphorylated somehow, and this helps to spread, I think. This is all.
Hi, here.
Yes.
Me or yeah? So thank you. This is beautiful work.
Thank
I
you.
really love it. So I have a question that is maybe I don't know if you will be able to answer, but I'm very curious to know what happened with the genome in these swarming cells, because I know Vibrio has 2 chromosomes, and there is a specific system that is anchoring the chromosome to the pole that is happy through the para-ABS system. And I'm wondering what happened in these swarming cells in which they are 10 times bigger.
Yeah. Okay. My collaborator, Ikro, saw the chromosomes in the longer cell. I think what he said there are many because the long swarming cell, the division is suppressed.
It's suppressed.
Yeah,
So
yeah.
they are polyploid.
Huh?
They have multiple chromosomes.
Yes.
Okay, and you have any idea, because you have a head, right, if I understood? So which is this pole? You know, which is the pole that is the head?
I see.
Yeah.
I see the point. Okay. The swimming organoleptical cell has one pole. This is checked by the TY localization. The most of the cell has one localized TY, but in our case there are 2. So the poles are 2. Yeah, both are 2.
Okay, can the head could be any of the pole, depending on the direction.
Yes.
Okay, that's excellent. Thank you.
Yes.
Yes. Okay. Okay. So, I must say I wasn't too surprised that the orientation of the bacteria is perpendicular to the
Yeah.
Because after all there is a water air interface. Right?
Yes.
And so given that there's a water air interface, I mean when it's turning,
Yes.
then the friction on one side is more than the friction on the other.
Yes, I think so.
And so that could also give rise to a turning.
Yes, I think so. Yeah, this is better than moving alone.
Yeah.
The both sides.
Exactly.
Yeah.
Yeah. But also it'll drive turning because the friction, this is the interface and the bacteria. The friction here is more than.
Yeah. Yes, I think.
There'll be an activity.
Is this the reason? You mean the chiral?
Yeah.
Yeah. Wait.
What I'm saying is that in addition to the they could also be this.
Yes.
Friction rate.
Yes. Actually, it's chiral. I didn't say in the presentation because it makes it a bit complicated, but yeah. Yeah, we can discuss it. Yes.
Hi, this was really, really beautiful work. And it comes back a little bit to what Linda was asking also. Have you considered the possibility of cell wall remodeling that may be coming because of EY localization or MREB changing its dynamic, which causes cell bending? It can then contribute to the swarming behavior you see.
I see. I have never considered, and my collaborator says something happened in the wild type cell, but at the moment, what we know at the moment is that we can measure the persistence length. It's quite flexible. In the case of wild type, maybe it's 4 micron or something. It's softer than the actin filament. And in the case for delta KY cell, it's double. It's maybe 8 micron. So from this, it looks the delta KY looks a bit rigid compared to the wild type. But only depletion of the KY affect. I have no clue about the relationship between the Murray or FTS and the key system. So it could be possible to consider other possibilities. But we should consider that. Thank you.
Okay, let's thank Masatoshi for reading.
Thank you very much.